DNA is the cornerstone of life, orchestrating the delicate dance of genetic instructions that govern living organisms. When the harmony of this biological symphony is disrupted through DNA damage, it can reverberate throughout the cell and ultimately affect the entire organism. Understanding the implications of this damage and the intricate cellular responses is critical to comprehending the fragility and resilience of life itself. Damage to DNA can arise from myriad sources, each posing a unique threat to genetic integrity. Environmental agents such as ultraviolet rays from the sun can scorch DNA, while ionizing radiation and chemical carcinogens stealthily inflict damage at a molecular level. Furthermore, during DNA replication, errors may creep in, leading to mutations that distort the genetic code. Reactive oxygen species, ROS, products of normal cellular metabolism, can oxidize DNA bases and fragment the essential phosphodiester bonds, while viral pathogens like HPV can invade and integrate into the host genome, further compounding the chaos. When DNA damage occurs, the stakes are incredibly high. Mutations triggered by damaged DNA can upset cellular equilibrium and pave the way for diseases such as cancer, where regulation of cell division spirals out of control. Aging processes are also intimately linked to DNA damage, contributing to the decline of cellular functions and the buildup of senescent cells that slow overall vitality. Moreover, DNA damage is implicated in neurodegenerative diseases like Alzheimer's and Parkinson's, showcasing its profound impact on the human condition. In response to DNA damage, cells activate an elaborate network of signaling pathways to orchestrate remediation efforts. Specialized proteins detect the breaches in DNA and send out a call to arms, mobilizing various signaling pathways that harmoniously coordinate the cellular response. The remarkable adaptability of cells is evidenced by their array of repair mechanisms, which include base excision repair, whereby damaged bases are replaced, and nucleotide excision repair, which cuts out and replaces the damaged DNA segment. In dire situations, if the damage is too severe for repair, cells may opt for apoptosis, a self-sacrificial act that prevents the propagation of irreparably damaged genes. Humanity's understanding of DNA damage and repair mechanisms is vital for advancing medicine, particularly in combating cancer and other age-related diseases. As we deepen our comprehension of these cellular processes, we inch closer to unlocking the mysteries of life itself, revealing the resilience inherent in our genetic architecture. By appreciating the delicate balance between DNA integrity and cellular response, we underscore the importance of safeguarding our genetic blueprint against the ravages of damage. Nucleotide excision repair. This pathway repairs damaged DNA by removing a section of DNA containing the damage and replacing it with a new section. Double-strand break repair. This pathway repairs breaks in both strands of DNA by sealing the break through a process called non-homologous end joining, NHEJ-4. Homologous recombination. This pathway repairs breaks in both strands of DNA by using a template with a similar sequence to repair the break conclusion. DNA damage is a common occurrence that can have severe consequences for the cell and the organism. However, the cell has evolved a complex network of signaling pathways and repair mechanisms to respond to DNA damage. Understanding the consequences of DNA damage and the mechanisms that have evolved to repair damaged DNA is essential for the development of therapeutic strategies to prevent and treat diseases associated with DNA damage.